Hey everyone, this video walkthrough will be covering OCR A-Level Biology Paper 1 from June 2024. If you do find this video useful, please do give it a like and subscribe. And a quick shout out to one of my subscribers for sending me the paper and making this video possible. First question is, which statement about lipids is correct? Lipids are polar molecules. That is not correct, they are non-polar. Lipids that contain fatty acids with carbon-carbon double bonds are liquid at room temperature. That is correct. The double bonds is what creates kinks in the chain. These kinks bend the chain and prevent them from coming close together, thus reducing the intermolecular forces present, which means it has a lower melting or boiling point, thus is a liquid. Now, I'm fully aware that the intermolecular forces part is chemistry, and if you don't do chemistry, I would still recommend that you know that part because it makes it easy to understand. Next option is not correct because saturated fatty acids do not contain the double bonds, and the presence of double bonds allow them to pack more closely. Well, that's not true because there's more kinks in the chain if they're double bonds. So again, the answer is B. Question 2 is lipids, polysaccharides, nucleic acids, and proteins are all macromolecules. Which statement about macromolecules is correct? So all macromolecules are formed in hydrolysis reaction. Not true, they're formed in condensation reactions. Lipids are not polymers, but polysaccharides, nucleic acids, and proteins are polymers. Yes, that is true. Lipids are not polymers because they are not made from repeating units of monomers. It's just one glycerol and 3 fatty acids. So that is the answer then. C is not true because lipids are not polymers, and not all macromolecules contain repeating units of monomers because lipids are the exception. Question 3 is the diagram shows part of a plasma membrane. How can molecule X be described? X looks to be a phospholipid, so it has a hydrophilic head and a hydrophobic tail. Yes, that is true. So the answer is A, but just to disregard the others, it's formed in glycerol and a triglyceride is replaced by a phosphate. Not true. It is when a fatty acid is replaced by a phosphate. It is non-polar. That is not true. It is polar due to the fact it has a hydrophilic head. The tails are joined to the head by peptide bonds. Absolutely not. Peptide bonds are only reserved for proteins. Question 4 is which statement describes a feature of the plasma membrane? So channel proteins are a type of intrinsic protein. That is true, so that would be the answer. Intrinsic proteins are proteins that span the entire length of the membrane. So this over here, as you can see, it's from the top to the bottom, so it spans the entire length, making it intrinsic. This structure over here is extrinsic because it's only on one half of the plasma membrane. So this looks to be something like glycoprotein, perhaps. So glycolipids are intrinsic proteins that have a lipid molecule attached. Not true. So it would be similar to what this structure is, and it's an extrinsic protein. So that should be extrinsic in order for it to be true. C is phospholipids form a rigid bilayer that membrane proteins are attached to. Not true. It is a fluid bilayer, and the fluidity is affected by temperature. Higher the temperature, the more fluid it becomes. D is that the plasma membrane forms an impermeable barrier. No, it's a semi-permeable membrane. Question 5 is adrenaline receptor is one class of a receptor known as a GPCR. The glucagon receptor on liver cells is another type of GPCR. Glucagon stimulates conversion of glycogen into glucose in liver cells. What is the action of glucagon? First option says that cyclic AMP catalyzes the conversion of glycogen to glucose. That is not true. Cyclic AMP is what you call a secondary messenger. So this is a secondary messenger model of action. The secondary messenger will in turn activate different enzymes that do the converting. It doesn't do the converting by itself. So that statement again, not true. B is glucagon is a secondary messenger. No, or a second messenger. Again, not true. Cyclic AMP is. Glucagon instead is known as a primary messenger. C is that the glucagon receptor is located in the cytoplasm of liver cells. Not true. It is located on the cell's surface membrane. And the way you can tell is due to the fact that glucagon is a peptide hormone, and these cannot pass through the bilayer. Only cholesterol-based hormones or steroid hormones can do this. D is when glucagon binds to its receptor, it stimulates the conversion of ATP to cyclic AMP. Yes, that is true, because the receptor is coupled with something known as adenylate cyclase, and that's what converts ATP to cyclic AMP. Now, this question is an application of knowledge question based on the theory that you had to memorize for the adrenaline receptor. I've taken this image from the textbook. It's basically how adrenaline works, very similar to how glucagon works, where both of them, glucagon and adrenaline, act as the first messenger. It binds and it activates adenylate cyclase, which does the converting of ATP into cyclic AMP, which is the second messenger, which in turn will activate other enzymes that convert glycogen to glucose. So they both work very similarly using this setup. Question 6 is lymph and tissue fluid are both formed from the blood. Which statement describes the composition of these fluids? Lymph contains more protein than tissue fluid because of antibody production. Yes, that is true, because lymph is where most of the immune cells reside, such as the B plasma cell, which is responsible for making antibodies, and antibodies are protein. So that statement is true, so A would be the answer. But just to disregard the others, So lymph is similar in composition to tissue fluid, but has more oxygen nutrients. Not true, because tissue fluid is formed from blood under high pressure, so when hydrostatic pressure is high, and that's coming from the arteries, which contains oxygenated blood. So tissue fluid would have more oxygen. C is tissue fluid does not contain hormones such as insulin and glucagon. That is not true. This is not true because how else would both these hormones actually reach the cells if not part of tissue fluid? And D is tissue fluid contains red blood cells and platelets. Not true either, because these things are too large to leave the capillaries. Question 7 is which statement describe features of the mammalian heart? Branches of the pulmonary artery supply blood to the heart muscle. Not true, it is branches of the aorta that do this. And also, pulmonary artery contains deoxygenated blood, so that would not be good to supply the heart muscle. The heart would die otherwise. Right, the next option is the semilunar valves prevent backflow of blood from the ventricles to the arteries. Not true, it is the the atrioventricular valves that do this. Instead, the semilunar valves would prevent backflow from the aorta or the pulmonary artery back into the veins. C is that the left ventricle pumps deoxygenated blood to the lungs. Not true, it is the right ventricle that does this. D is that the wall of the left ventricle is thicker because it needs to pump blood around the whole body. Yes, the thickness of the wall is what generates the pressure, and by process of elimination, D would have to be the answer. Question 8 is bumblebees are large insects that have a high demand for oxygen during flight. What adaptation enables bumblebees to obtain sufficient oxygen during flight? Accumulation of lactate in muscles during flight increases the amount of tracheal fluid, which increases the surface area for gas exchange. Now, this statement sounds contradictory because lactate is in muscles. So if it's in muscles, I don't see how that would increase tissue fluid unless it leaves. But the statement just seems wrong altogether because fluid does not increase surface area. So that entire statement is just off. B is that muscle contraction before or during flight increases ventilation. Well, yes, that would increase ventilation because you're opening and closing the spiracles that way. So yeah, the answer would be B. So C is incorrect because spiracles would take it to the body cavity, not the tissue, and tracheals do not open and close because spiracles can open and close. 9 is which option is not an adaptation? So not an adaptation that helps fish increase the efficiency of gas exchange. So blood capillaries lie close to the surface of the lamellae to minimize diffusion distance. Well, that is true, so it's not the answer. Blood flows in the capillaries in the same direction as the flow of water over the lamellae to maximize gas exchange. That is not true. Blood and water flow in opposite directions. This is known as the counter current system or the counter current multiplier. So because it's not true, it is the answer. The other two would be correct, therefore not the answers. The next question shows a diagram of a liver lobule. Which statement about liver function is correct? Now, before we go through this, I just want to annotate the diagram a little bit. So I is the hepatic vein, and the way you can tell is due to the fact that it has the largest diameter, which is a feature of the hepatic vein. E is known as a sinusoid. That is where the blood from the hepatic artery and the hepatic portal vein mix together. H over here is the hepatic artery, and the way you can tell is because it has a smaller diameter than G, which happens to be the hepatic portal vein. And lastly, F is the bile duct. Yep, that's a bile duct, and this in the middle is known as the canaliculi. Sorry, that's not spelled right. Canaliculi. That is where the bile is collected for when it drains into the bile duct. So now that we know that, the question is which statement about liver function is correct? So blood flows from I towards G, I towards G. Yep, not true. Blood would flow in the opposite direction, from G towards I, because the G is the hepatic portal vein blood, which drains into the hepatic vein. The next option is H carries blood arriving from the digestive system. We said H was a hepatic artery, so that is not true. This blood comes from the heart. It's the hepatic portal vein that collects the blood from the digestive system to the liver. So yeah, that's not true. Hepatocytes produce urea that flow into F, and F was the bile duct. That is not true. It's the bile that flows into F. Urea flows into the vein. So not the answer. So that means D would have to be our answer, which is toxic substances enter liver cells from the blood at E. So E, again, is a sinusoid where the blood from G, which is the portal vein, and H, the artery, mix together and they enter this region. Then once the liver cells have finished breaking down alcohol, different medications, food, etc., those things enter into the sinusoid and then drain into the vein, which takes it to the kidneys for that to be excreted and then sends it back to the heart. Question 11 is a photomicrograph which shows a stained section of liver tissue. What structure is labelled J? So before we even start that, just annotating other structures. So we have this over here, the largest lumen that we could see. This is the hepatic portal vein. Sorry, not portal vein, just the hepatic vein. And we know this from the previous question. But the thing with the largest lumen diameter is the vein. Then as for the other structures, well, we see this over here. It has a small lumen. Well, yeah, this has a small lumen than this. The thing with the smallest lumen is the hepatic artery. Slightly larger is the hepatic portal vein. So the answer to this question then is B, hepatic portal vein. Question 12 is which statement describes urea production in the liver? Amino acids enter the ornithine cycle. Absolutely not. It is ammonia that enters the ornithine cycle. This is just something you have to memorize. Ammonia and carbon dioxide combine to make urea in the Krebs cycle. Well, that's true. They do combine to make urea, but not in the Krebs cycle, in the ornithine cycle. Krebs cycle is respiration. Ammonia is produced by the deamination of amino acids. Yes, that is true. So by removing the amino group or the amine group, that is what makes ammonia. So the answer is C. The last one says that ornithine cycle makes urea less harmful. That is not true. It makes ammonia less harmful. Urea is the less harmful product. Question 13 is which statement about the effect of light intensity on the concentrations of GP, RUBP, and TP is or are correct? Statement 1 is at low light intensities, less GP is converted to TP because there are less products of the light-dependent stage available. Yes, that is true, because we need products of the dependent stage to convert GP to TP, those products being NADPH and ATP. The second statement is at high light intensity, RUBP concentration is high because it is regenerated from TP. Yes, that is correct, because high light intensity would produce said NADPH and ATP, which allows TP to be made, and in turn TP will regenerate RUBP. The last statement is at high light intensity, RUBP accumulates because it cannot be converted to GP. Well, this is not necessarily true, because in order to be converted to GP, it needs carbon dioxide, and carbon dioxide is not a product of the light-dependent stage, so it is not dependent on the light intensity. So those two things just do not correlate, which is why the third statement is incorrect. So the answer would be B. Question 14 is which of the statements about factors affecting the rate of photosynthesis is or are correct? First statement is lack of water is not usually a limiting factor, although it does cause the closure of stomata, which reduces carbon dioxide levels. Now, you would think that this is true because at GCSE level, they tried to hammer home the fact that water is a limiting factor because it's one of the reactants in the overall reaction of photosynthesis. But it is more nuanced than that, and you know this because of cyclic and non-cyclic photophosphorylation. If you think about non-cyclic photophosphorylation, that process requires water because water will be broken down by photolysis. So photolysis of water provides the electrons that replace the electrons that were lost from the photosystem. That is when water is needed, but it is not needed if it's cyclic photophosphorylation, because in cyclic, the electrons are returned back to the photosystem. So water does not play a role in that. Therefore, it is not usually a limiting factor, which makes the statement true. The second statement is carbon dioxide concentrations can be low inside greenhouses, therefore growers will often enrich the atmosphere with carbon dioxide. Yes, that can be done because greenhouses are made so that you can control such factors. The third one is that between 0 and 25 degrees Celsius, the rate of photosynthesis doubles every 10 degrees Celsius rise in temperature. Now, I've searched high and low to see if the statement's true. It's not in the textbook, not the specification, not in any other OCR resources I've seen online. So I'm not sure where they're getting this fact from. If you do know where, can you please put it in the comments, because I'm genuinely curious, because I genuinely can't seem to find anything online. But then it got me thinking that I might be reading too literally into the statement. I think what they're trying to get across over here is the fact that temperature is one of the limiting factors, and by increasing temperature to a certain point, it increases the rate of photosynthesis. I think it just might be that simple of a statement that I may be reading too much into. But if you are aware of this, please do tell me. Right, so all three are true in that case, so the answer is A. Right, so then the last question over here is which option about the relative energy values of different foods is not correct? So carbohydrates have lower energy values because they have a higher oxygen content. Yep, that is partially correct. So it's lower than lipids, but higher than proteins, really. And the reason for this is because of the high oxygen content. So what gives something a higher energy content is the presence of carbon to hydrogen bonds. Because when these are oxidized, it is extremely exothermic, so releases a lot more energy. Carbon-oxygen bonds, yeah, they're not the same as this, right? So we want more carbon-hydrogen bonds. So that is a true statement, therefore not the answer. Food that has a mixture of carbohydrate, lipid, and protein, such as chocolate, has a higher energy value than the same mass of sugar. Yes, that is true because the presence of lipid supplies the carbon-hydrogen bonds. Lipids have energy values lower than proteins. Well, that is not true because lipids have more carbon-hydrogen bonds, so they should have higher energy values. Therefore, that is not correct and is the answer. So the answer is C. The last statement is proteins and carbohydrates having similar energy values. Yes, that would be true. because of the oxygen content present. Section B, question 16, shows a table with some biological molecules. Complete the table by putting a tick in the appropriate box or boxes on each line to show whether the corresponding feature is present. The first line has been completed for you. So the next thing is amylopectin, which is a polymer made up of glucose monomers, and it does contain glycosidic bonds. Glucose is just a monomer, so no glycosidic bonds. Sucrose is a disaccharide made up of glucose and fructose monomers. So it's not a monomer because it's a disaccharide, not a polymer either because that implies more than 2 repeat units, but it does have glycosidic bonds. Describe the bond between two glucose monomers in maltose. So it would be two alpha glucoses bonded together. Therefore, this bond is called alpha. Now, you don't need the alpha for the answer, but it's an alpha 1-4 glycosidic bond. So that is 1 mark for carbons 1 and 4, second mark for the glycosidic bond. Question C is maltotriose is a trisaccharide formed during the breakdown of starch by amylase. It can be broken down further to produce glucose. Complete the equation for the conversion of maltotriose to glucose. So in order to break this down, we have to hydrolyze 2 glycosidic bonds, therefore requires 2 water molecules. The enzyme maltase converts maltose to glucose during the final stages of starch digestion in the small intestine. So just why maltriose can be converted to glucose by maltase. So maltase works specifically on maltose, but it can also work on this substrate, which should tell you that both substrates are complementary to maltase's active site. If you will be writing the first statement, you do have to clearly state active site in order to score the mark. In the next question, figure 17.1 shows a relationship between photosynthesis and aerobic respiration. Identify the molecules labeled X, Y, and Z in figure 17.1. So X looks to be a product of aerobic respiration that is used in photosynthesis, specifically the light-dependent stage, and that would be water. Y looks to be a product of aerobic respiration, but used in the light-independent stage, and that will be CO2. And Z looks to be a product of the light-dependent states that's used in aerobic respiration. So we already have glucose, the thing that's missing is oxygen. The next question is to use information in figure 17.1 to explain how plants are able to survive in a sealed glass container such as a terrarium or a bottle garden for many months or even years. So looking at the cycle, it looks to be like the products of one reaction happen to be the reactants of the next, and it continuously goes on. And that is pretty much the answer, except we have to be a bit more specific. So you could say that respiration produces carbon dioxide and water, which are both reactants in photosynthesis, and then photosynthesis produces glucose and oxygen, which are both reactants in aerobic respiration. And the only other thing that is outside of the terrarium is light energy, but if it's a glass bottle, then that is easily accessible. And that's pretty much it for 3 marks. I will also list the other possible points that were on the mark scheme. These are all of the indicative points, but of course you only need 3 of them. And as for these 2 points where it says name celled activities or name mineral ions, you do have to state an example. So for mineral ions, popular ones include like an ammonium ion, nitrite ion, anything like that, or even like magnesium, etc. And then for the cell activities, the go-to answers are usually protein synthesis or something like cell division. Question B is algae such as seaweed can occur in a range of colors. Algae carry out photosynthesis in the same way as plants, using chlorophyll A as the primary photosynthetic pigment. Figure 17.2 shows the percentage of light at each wavelength that is used in photosynthesis by the red alga by this name. I'm not going to pronounce it, otherwise I will butcher it. Besides the point, chlorophyll A has an absorption peak at about 618 nanometers. Use figure 17.2 to estimate the percentage of light absorbed by chlorophyll A that is used in photosynthesis by this species. So pretty straightforward, we can see the percentage of light used in photosynthesis. So all we have to do is go to 680 and then sort of see where it crosses that. So by the looks of it, it may seem like over here, which is 20, but not really, because if I zoom in even more, it actually crosses at this point, which is 22. So that's what the answer is. The answer is 22. Other correct answers would have been 21, 23, or 24, but the answer, which is just 20, is just not on the mark scheme. The next question is to explain why the percentage of light used in photosynthesis is higher than your answer to part I at wavelengths other than 680 nanometers. So looking back at the graph, I believe they're referring to this section, which is where the light absorption peaks. And the reason why it can absorb wavelengths in this range is due to accessory pigments. So it tells us that chlorophyll A has this absorption peak, but chlorophyll A is known as a primary pigment or the reaction center. By having accessory pigments present, it allows a wider range of wavelengths to be absorbed, and these pigments, when they absorb the light energy, they allow it to be funneled down to the reaction center or the primary pigment. So that's pretty much what the answer is. In the next question, we're told that this species grows in deep water. Use the data in figure 17.2 to suggest how it is able to survive in conditions where other types of algae and plant cannot. So, looking back at the data, it looks like its peak absorption is at this wavelength, which is a lot shorter than the chlorophyll A's peak, which is 680. And the reason why this would be an advantage is due to the fact that this plant lives underwater. So if light's going to be going through the water to reach the plant, it is beneficial to have pigments that can absorb the shorter wavelengths. Reason being, shorter wavelengths can penetrate waters at greater depth. Now, that is bordering physics, really, which is why it's an advantage to do all three sciences at A level. But besides the point, if you did not know that already, I would highly recommend you put it in your notes or your flashcards so that you're aware that this could be a point or something that could be asked of you. Question C is Melvin Calvin and co-workers worked out the reactions of the light-independent stage of photosynthesis. They illuminated a suspension of green algae in the presence of radioactive carbon dioxide. They removed samples of the suspension at different times after addition of the radioactive carbon dioxide and used paper chromatography to identify the compounds in what is now known as a Calvin cycle. Figure 17.3 shows their results. Calvin concluded that GP was the first product of the carbon fixation process and that GP was converted to TP. Use data in figure 17.3 to explain how Calvin reached this conclusion. So again, we're looking at GP being first and then that being converted to TP. So looking at the results, we could see that yes, in fact, GP does form first because it forms after the first second, and it's only after 5 seconds that triose phosphate is formed. Next question is to state what you can conclude from figure 17.3 about what happens to TP. Looking at the figure, we can see that the triose phosphate goes on to form RUBP, sugar phosphate, and then after 30 seconds it also makes glycine, serine, and other amino acids. So the answer is that it's converted into sugar phosphates, and sugar phosphates named amino acids. So again, that would be specific, such as, what was it again? Yes, glycine or serine, you could name those, as well as RUBP. And they also said sucrose, yeah, which is present over here. So sucrose as well. Question 18a, furosemide is a type of drug known as a diuretic that acts on the nephron to decrease reabsorption of water in the collecting ducts. Diuretics are often prescribed to treat high blood pressure. Furosemide is on the list of banned substances published by the International Olympic Committee. State one other example or type of drug on the list of banned substances that can be detected in urine samples. Common example would be steroid hormones. Other examples include cannabis, as well as stimulants like amphetamine. Right, the next question is, furosemide can also be misused by jockeys or boxers who need to be below a certain weight. Explain how diuretic could help reduce weight. So diuretics cause you to want to urinate more. So by losing lots of water, that would decrease body mass. But the question is mainly to explain how. So what furosemide would do is to make the collecting duct more permeable to water. And as a result, there's going to be rapid water loss. And as a result, less weight. Furosemide is banned substance because it can be used as a masking agent to hide the use of performance-enhancing drugs. So just how furosemide can act as a masking agent, the waste products of the enhancing drugs is excreted to the urine. So by using this diuretic, it increases the volume of urine, therefore most of the leftover drug particulates leave the body, making it undetectable. Question B is, urine analysis can also be used in medical diagnosis. Bladder cancer can be diagnosed by surgical removal of a small piece of bladder tissue called a biopsy sample. State one advantage of urine analysis over biopsy samples. So, well, it doesn't require surgery, obviously, so less painful, less invasive, less infections, that sort of thing. Also a lot cheaper, and it's also more rapid. Your testing a urine sample would be a lot more rapid than going through the whole surgical procedure. Question II is to give another application of urine analysis and diagnosis, state the substance measured and the corresponding medical condition. So a good example would be diabetes. Sorry, that's not the substance. That's the medical condition. The substance is glucose, and the medical condition is diabetes. Another one could be protein in the urine. That is indicative of kidney damage. Also like creatine, indicative of muscle damage or kidney damage as well. The formation of tissue fluid has many similarities with the process of ultrafiltration in the kidneys. The table below shows factors involved in the formation of tissue fluid and its return to the blood. The net pressure at the arterial end is positive 1.3 kilopascals. Calculate the net pressure at the venual end. So then to calculate the total pressure or the net pressure, we want to find the differences in pressures of the hydrostatic and oncotic pressure. And then within this, you also want to find the net hydrostatic pressure to subtract it from the net oncotic pressure. So what I mean is for hydrostatic pressure, the total pressure at the venual end is 1.7 subtracted by 1.1. That is equal to 0.6. Now, the reason why subtracting them is because blood pressure is forcing it outwards, but the tissue fluid pressure on the other end is pushing it back into the blood. So it's almost like playing tug of war, but more like pushing rather than pulling. So we want to find the difference to find out the overall force of the hydrostatic pressure. So subtracting it. A very similar thing for the oncotic pressure. We need to subtract both values. So 3.3 subtracted by negative 1.2, and that is negative 2.1. Then we want to add both of these pressures to give us the net pressure, or the total pressure. So that would be 0.6 plus negative 2.1. That's a total of negative 1.5 kilopascals. The next question is to use the information in the table to explain the formation of tissue fluid and its return back to the blood. So, just diagrammatically explaining it first in case you've forgotten this from AS. So here's what the capillary looks like. We've got the arterial end and then the venual end. So the arterial end has blood coming under higher pressure due to the contraction of the left ventricle. So the high pressure that's generated from the left ventricle is known as the hydrostatic pressure. So the high hydrostatic pressure is greater than the oncotic pressure, so lower oncotic pressure, and you can prove this to yourself by finding out the total oncotic pressure at the arterial end later if you fancy. But basically, the high hydrostatic pressure is what forces the fluid out, so that fluid being tissue fluid, which is comprised of water, plasma, glucose, amino acids, oxygen, the small stuff, basically. Now, because all of the water or the fluid is leaving, that leaves behind proteins in the blood. These proteins are like albumin, or just larger protein type of material, which lowers the water potential. This low water potential is the same thing as saying a high oncotic pressure. So oncotic pressure is the pressure due to proteins that remain in the blood. So the high oncotic pressure, or if you fancy, the lower water potential, is what draws the fluid back in at the venual end. And I forgot to mention this earlier, but whenever oncotic pressure is high, hydrostatic pressure is automatically low. Whenever hydrostatic is high, oncotic pressure is low. They just work in opposite ways. So that would be the second mark, the fact that hydrostatic pressure is lower than oncotic pressure, therefore fluid moves back in at the venual end. The next question is to describe the homeostatic mechanisms that regulate the water content of the blood by changing how much water is reabsorbed from the collecting duct. You do not need to describe how the loop of Henle sets up the water potential gradient in your answer. Now, this question is marked by an asterisk, so it's assessing the quality of written communication, and it is also 6 marks. This diagram that I've taken from the textbook shows the role of the hypothalamus and the posterior pituitary in the production and secretion of ADH, which is the antidiuretic hormone, and is a key hormone in regulating the water content. So we start at the osmoreceptors, which detect the water potential of the blood. And when they detect the water potential, depending on whether it's high or low, the cells will tend to shrivel up or swell up. Now, let's go with a low water potential. So if this is detected, the osmoreceptors will shrivel and in turn will kickstart the production of ADH. ADH, once it's produced, will get stored in vesicles inside the posterior, specifically the posterior pituitary. Once it's over here, it gets released into the bloodstream, and then it goes towards the kidneys, specifically through to the collecting duct of the nephron. And this down here shows what happens next. So in the middle, we have the lumen of the collecting duct, which is through where the filtrate goes through. And these blocks or cells that you can see at the sides are the cells that line the collecting duct. So ADH, which is a peptide-based hormone, will bind to its receptor on the surface of the cells that line the collecting duct. And then this will trigger the secondary messenger model cascade of a reaction. You don't have to go into the details of the reaction, but it's good to mention considering it's marked with an asterisk, so quality of written communication. Well, that's besides the point. So ADH will bind, secondary messenger model happens, and these vesicles containing aquaporins, so that word is aquaporin. So aquaporins are just water channel proteins that get embedded into the collecting duct membrane. So these vesicles containing the aquaporins will fuse with the membrane and result in the embedding of these water channels in the membrane. So the water that's coming through will enter these cells through the aquaporins. And that is pretty much it for the 6 marks. I'll put in a picture of what the mark scheme has detailed, so you can have a look for yourself. Question 19a is plants respond to their environments in different ways. Describe the meaning of the term tropism. So this is the directional growth in response to a stimulus. growth in response to a stimulus. So it's just one of those definitions you just have to memorize. The next question is to name one type of tropism. So you could have said phototropism. So this is in response to light. Geotropism, which is in response to gravity. And hydrotropism, which is in response to water. Just to name a few. Others that were accepted were thigmotropism as well as chemotropism. Give one example of an abiotic stress and the corresponding plant response. So stresses, so abiotic are the non-living factors that cause stress, so like lack of water, lack of carbon dioxide, pH, temperature, etc. So let's go with lack of water, for example. Lack of water, that results in the closing of stomata. So to preserve water and prevent transpiration, closing stomata. Another example could be high temperature, and this would also close the stomata. Close stomata to prevent water loss and cause dehydration. Another thing could be like high wind speeds, and again, you could say closing stomata for that too. With the low light levels, if you were to say that, you could have described abscission, which is just leaf 4. Question B are mycorrhizae are associations between some types of fungi and the roots of plants, including trees. The fungal hyphae grow into the roots and help the trees to take up water and minerals. Trees respond to insect attacks by producing chemicals that defend against insects. Trees also release pheromones into the air that stimulate nearby trees to produce defensive chemicals in preparation for possible insect attack. It is known that communication between trees is of the same or different species can also be carried out via mycorrhizae, which were, the fungal stuff. Okay. Some scientists investigate this phenomenon using young fir and pine plants. This is the method they use. So they grow a donor fir plant together with a recipient pine plant in the same large pot, repeat to create 40 pairs of plants, divide the plants into 4 groups with 10 pairs of plants in each group, enclose the roots of the recipient plants of each pair in a mesh bag, stimulate insect herbivory by removing the leaves of donor plants in half of the groups. The treatment groups are summarized in the table, and scientists measure the concentration of the enzyme polyphenol oxidase PPO in the recipient seedlings at the start of the experiment and after 72 hours. PPO is involved in the production of defense chemicals. So just how the scientists could ensure that airborne pheromones did not contribute to communication between plants in this investigation. So prevent airborne pheromones, well, if they're in the same pot, so they're close together, so we can't move them apart now. Were they in the same pot? Yes, they were put in the same large pot. So we can't move them further apart then, but we could prevent airflow between them by like placing a plastic bag perhaps on top of it. So you could you could enclose the plant in an airtight bag of sorts, so airtight plastic bag. The results are shown in this table, so we could see the mean PPO activity plus minus standard deviation in arbitrary units at the start and at the end, and for each of the groups as well. So the scientists concluded that removing the leaves of the donor plants leads to a chemical defense response in the recipients. This is due to signals carried by the mycorrhizal hyphae. Evaluate the support given by the results to the scientist's conclusion. You should comment on the quality of the scientist's data in your answer. So just to reiterate, the scientist's conclusions is that removal of leaves leads to increase in chemical defense secretion in the recipients, and this is due to the mycorrhizae hyphae, so due to the fungus. So looking back at the tables, so analyzing what they did first, we could block this off as 2 separate groups. So groups 1 and 2 had the smaller mesh size that only allowed passage of solutes, but not the roots and the hyphae, and then 3 and 4 had a large mesh size which allowed movement of solutes and hyphae, but not of the roots. So comparing groups 1 and 2 in terms of results, so this one had leaves removed, so removed over here, and they were removed over here as well. Okay, so comparing groups 1 and 2, it looks like at the start the levels are pretty similar because the standard deviations overlap. And after the 72 hours, it looks like after the leaves have been removed, there's a greater increase in the PPO, which is the phenol oxidase stuff. Okay, so that shows us that removal of leaves did have an effect, so this correlates with the scientist's conclusion. The next thing was it's due to the mycorrhizae. And this is when, okay, so now we're looking at 3 and 4, which involved a larger mesh size, therefore passage of the hyphae. So comparing 3 and 4 then, we could see that again at the start, levels are similar but not exactly the same because standard deviations do not overlap. So that could contribute to the results not being as valid because they did not find a way to absolutely control the starting concentrations. Okay, that's just something to keep in mind. Besides the point. Point being that there's a very large increase after 72 hours with the leaves removed and by allowing the hyphae to passage between the 2 plants. So both these points would be in support of the scientist's conclusion. Now, in terms of not supporting the scientist, looking at the results where the leaves were not removed, there still seems to be an increase in the PPO. So this one's not a marginal increase because standard deviations do overlap, but with number 3 over here, there is a pretty big increase. So that goes against the scientist's conclusions. So then this would show that leaf removal does not technically have an effect, because, yeah, as for these two, and also the hyphae passage does not affect it really because it seems to have increased without removing the leaves, as well as allowing the hyphae to passage between both crops. So yeah, that seems to be about like 2 marks for supporting and then 2 marks for not supporting the scientist. But then we also have to comment on the quality of the data. So it looks like they've done standard deviation, which is great, because then this tells us the deviation from the mean, which is good. However, it would be better if they did an actual statistical test, like a t-test, to compare significance between the means. Another thing is these conclusions are quite broad, because it just says removal of leaves in general, right? But we've only used 2 types of crops, trees, if I remember correctly, yeah. It's only just the young fir and the pine plants, so this information is not translatable to other types of trees, which would decrease the validity, I would say, of the scientist's results. Question 20 is, students investigated ventilation and heart rate in a resting subject. The subject breathed in and out through a spirometer. The spirometer chamber was filled with room air. Soda lime was present to absorb any carbon dioxide. The results are shown in figure 20.1. Okay, first question is that the downward slope of the spirometer trace in figure 20.1 is due to oxygen consumption. The subject exhales the same volume of air as they inhale. So the volume exhaled is the same as inhaled. We need to state why the trace slopes downwards. Now, this question's quite odd because they tell us the answer, oxygen consumption, right? So if you look at the start, the volume of air is greater than the volume after because oxygen is used by cells. Very odd question, but that's what the answer on the mark scheme is that oxygen is used or it's being removed. Now, I'm going to be honest with you, I don't think I would have ever thought of this in an exam, considering it's already stated, so my mind would be, what else? But that's not what the mark scheme says. Actually, the other accepted answers were that carbon dioxide is being absorbed. I think that makes more sense, because we have soda lime present. So what's happening is you're exhaling, so you inhale the oxygen, exhale the carbon dioxide, then that carbon dioxide is absorbed, which is why the volume decreases. I think that's a much better answer, but that's on the allow section. The next question is that the subject had a mass of 75 kilograms. Calculate the subject's oxygen consumption in centimeters cubed per kilogram during the first 2 minutes of the experiment. So we're looking at oxygen consumption, therefore a change in the volume of the tank or the container initially versus at 2 minutes. So if it's centimeters cubed per kilogram, it would be the volume divided by the kilograms, which is 75. So on the trace at 2 minutes, that's what it is, about 0.3, give or take. Yeah, I think you go slightly higher than that, maybe 0.4. And then at time 0, it's about 1.5. So you could do 1.5 subtracted by 0.3, which is equal to 1.2 decimeters cubed, but they wanted it in centimeters. We could divide this by 1,000. And then per kilogram is divide all of this by 75 kilograms. Now, here's a quick catch, is this value that I told you, which is 1.2, that is actually outside of the range in the mark scheme, because the range of volume in the mark scheme that was accepted is between 0.9 to 1.1. And if I'm going to be honest with you, it doesn't look like that would be the volumes, because here's 2 minutes, which, yes, is 120 seconds. That's where the bottom peak lies, and then the top peak is actually up, yeah, around 1.5, you could say. 1.6, if we're being technical. So that difference is really not in the range of 0.9 to 1.1. If you do have a slightly different approach to this, feel free to put it in the comments, but this is the only way I can think of answering the question. But in this way, it falls out of the mark scheme bounds, or the range in the mark scheme. However, you would still get marks for this part, which is dividing it by 75. And as well, yeah, just 1 mark considering it's outside the range, but you also get marked for the 2 significant figures. So then the next question is to use the figure to compare breathing pattern between 0 to 120 seconds and 120 to 240 seconds. So then looking back at the trace initially, so 0 to 120, if you can see these curves, just zooming in here, so if we could see these curves, that up and downness is known as the amplitude. The amplitude looks quite small because after 120 seconds, yeah, this is how big the amplitudes get. Right, so the bigger the amplitude means deeper breaths have been taking place. So you could say that initially the breathing rate is pretty shallow, or the breath depth is pretty shallow, and then that starts to increase 120 seconds onwards. So the answer to the question is at 120 to 240, there's deeper, so that's the mark for saying deeper breaths. And we can tell because the amplitude or tidal volume is greater, or you could say more variable. The next question is the student measures the subject's heart rate and blood oxygen saturation every 30 seconds. The results are shown in figure 20.2. So we've got a key, the non-shaded circles are heart rate, shaded circles are oxygen. Use the data in figure 20.1 and 20.2 and your own knowledge of homeostatic control to explain the changes in breathing rate and heart rate during this experiment. So in this experiment, it doesn't seem like they're doing any exercise, not that they have mentioned it. I believe they were just at rest initially, as stated by, yeah, it's just a resting subject. Okay, so what they have done is the subject is still at rest and we're measuring heart rate and oxygen saturation for every 30 seconds. Now for the heart rate, we can see that as time increases, there's a sort of jagged but still increase in heart rate. So we know that for a fact is heart rate increases. What about breathing rate? Now the oxygen saturation decreases. So that means that to replenish the oxygen, the breathing rate should have increased. So the first mark is to say that the heart rate increases and the breathing rate also increases. Now that we know that, we have to state, using our knowledge of homeostatic control, so why would heart rate and breathing rate increase to balance the internal conditions, which is homeostasis? So if oxygen concentration is decreasing, that automatically would tell us that carbon dioxide is increasing, because all of the cells take up the oxygen, use it in respiration, and excrete out carbon dioxide. Carbon dioxide would make the blood pH decrease. So the oxygen saturation decreases, and as a result, the blood pH increases. And it increases because carbon dioxide reacts with water in the plasma to make carbonic acid. Right, and you should remember from previous AS content that this the enzyme that catalyzes this reaction is carbonic anhydrase. Besides the point. Okay, so the blood pH decreases, and this change in blood pH is detected by specific receptors, those being chemoreceptors. So chemoreceptors in the medulla. So chemoreceptors in the medulla oblongata. They would detect change in pH, and in turn, it would stimulate the heart rate to increase. Right, so the stimulus goes to the sinoatrial node, and to speed up the heart rate, that is through the sympathetic nervous system response. So impulses, so more impulses along sympathetic nerves to the SAN. Right, and the SAN would respond by increasing its rate at which it produces a wave of excitations, and thus the heartbeat. Now this is way too many points for 4 marks, so summarize it as you will, but each bullet point over here is worth 1 mark. The next question is blood oxygen saturation less than 90% can be dangerous. Explain one modification you could make to the experiment to safely study the effect of extended periods of exercise. So if I remember correctly, the experiment that they had done, it's over here, is yes, the spirometer chamber was filled with room air, and then this person or the subject keeps breathing in and out this air in the chamber. So the air is not changed in the chamber, that air in the chamber remains constant. Now, if this air remained constant, remember, more carbon dioxide is produced, but that is absorbed by lime water. But at the same time, oxygen keeps getting consumed, so the oxygen in the tank would decrease. Now, if oxygen in the tank decreases, then that means that the blood saturation would decrease. So to make it safe, you'd want to increase the oxygen content in the spirometer, so to enrich the spirometer with oxygen so that the blood oxygen saturation is constantly maintained. Question C is figure 20.3 shows how the respiratory quotient of an athlete changed during exercise from rest up to 100% maximum effort. State 2 measurements that you would need to make during the experiment to calculate RQ. Now RQ, the respiratory quotient, is the ratio of oxygen used and the amount of carbon dioxide produced. Therefore, to calculate it, you need both of those. So that's volume of oxygen used, or you could have said inhaled. And the second thing is the volume of carbon dioxide exhaled or produced. Next question is to explain what figure 20.3 shows about the respiratory substrates used at different intensities of exercise. Now, this question is related to respiration, where you have to memorize the RQ values associated with certain food types, such as lipids, which is about 0.7. Again, you just have to memorize these. proteins are about 0.9, and carbs are about 1. And the reason why carbs have a respiratory quotient of 1 is because if you think back to the aerobic respiration equation, the balanced equation, it's 6 oxygens that are used and 6 carbon dioxides that are made. So the ratio of oxygen and carbon dioxide are equal, therefore 1 is a respiratory quotient. Now, as for the other two, when it comes to analyzing it, proteins are never the go-to be respired. Reason being, proteins are extremely important. They're used in cell microtubules, cytoskeleton, enzymes, actin, myosin, lot of structural things that if you were to consume that for food, the body would disintegrate. Not ideal. So proteins are the very last resort. Lipids is what the second go-to is. If you don't have carbs, lipids would be your best friend. And if lipids run out, that's when the body would have to rely on proteins. But again, that is a very last resort. The reason why I'm telling you this is because this could come in handy for your revision if this sort of question shows up in your actual exams. Besides the point though, so looking back at the question, what figure 20.3 showed about the substrates at different intensities. So the two intensities mentioned were 0 and 100. So at 0, we see 0.7 is the RQ value. So we could say that at rest, lipids are the go-to substrate for respiration. And at 100, it is, oh, okay, that's pretty high. So bring that slightly lower, about 95. Now, not proteins, because proteins is 0.9, not 0.95. So this is actually carbs, because remember, like I said earlier, proteins are the very last resort. Following question is 21a. Most digestive enzymes are extracellular. Complete the sentences about the synthesis and secretion of a typical digestive enzyme using the most appropriate terms. The polypeptide chain is assembled at ribosomes on the rough endoplasmic reticulum. So on the mark scheme, you could have abbreviated it or written out the entire phrase. Then after this, the protein is transported to the Golgi, so the Golgi body or Golgi apparatus, where further processing occurs. The final protein is packaged into vesicles for transport to the plasma membrane, where the protein is released by exocytosis. Hydrogen peroxide is a highly reactive chemical. Catalase is an intracellular enzyme that catalyzes the breakdown of hydrogen peroxide. Explain why it is important that catalase is able to break down hydrogen peroxide very quickly. So this question is sort of referring to like a case study, if you want to call it that, of the catalase enzyme. So you need to know that catalase breaks down hydrogen peroxide into water and oxygen, which are both harmless products, whereas hydrogen peroxide is not harmless, it's actually very toxic. So they're avoiding to write the word toxic over here because that would actually be the answer. So hydrogen peroxide is very toxic, and the fact that the products are harmless. Products, that being water and oxygen. Yeah, you do need to state that, so I don't mind putting that in brackets. So water and oxygen are harmless. Right, if you're a bit confused as to how they're pulling this from thin air, they're not. This is something you just have to memorize. It is in the specification. This is like a need-to-know example. Part 2 is figure 20.1 is a graph of decomposition of hydrogen peroxide against time for catalase. So here we got hydrogen peroxide decomposition in micrograms on the y-axis, time on the x-axis. Calculate the rate of enzyme action at 30 seconds. So 30 seconds is about here. We have to draw a tangent as this part of the graph is curved. Now on the screen of a tablet, it's very difficult to draw tangents. But yeah, just use a ruler and try to align it accordingly. That looks atrocious. Also, quick tip is when you're doing tangents, make sure that the tangent is large enough because that reduces the uncertainty of the gradient when you calculate it. So you want the tangent to cross through both the y-axis as well as up here, because the greater the tangent, the smaller the uncertainty of your answer. The smaller your gradient, the uncertainty increases, which just means you're more likely to make a mistake. But anyway, this tangent still seems atrocious considering it's not just touching that point, but you get the gist. You'd work out the gradient of the tangent, and that would be the answer. So then the answer that's on the mark scheme is 3.7 plus or minus 0.6, accordingly. I would calculate it, but I can't seem to draw the tangent correctly on here. And then the units for this is micrograms per second, because we're dividing peroxide by time. Question C is male infertility is associated with low mobility, so ability to move, of sperm cells. Superoxide dimutase, SOD, is an enzyme that is often located together with catalase in cells. Superoxide ions are produced in mitochondria and are highly reactive. Superoxide ions cause damage to biological molecules, including DNA and lipids. SOD converts superoxide ions into hydrogen peroxide, and oxygen, explained by sperm cells, might have a high concentration of hydrogen peroxide. Well, they are sperm cells, and one of their key functions is to migrate to the egg cell, so they require to generate energy. Energy comes from mitochondria, so the reason why it would be high is due to having many mitochondria, for one. Another reason could be the fact that catalase activity is low. Catalase activity is low. Now, how would you know it's low? Couldn't tell you, but that's what's on the mark scheme. However, I do have a feeling it's due to the fact that SOD's rate of conversion of superoxides is extremely high, which means that the concentration of hydrogen peroxide at any given moment is extremely high, making the number of catalase enzymes the limiting factor, because there's just way too many substrates. So that's why you could say catalase activity is low. Honestly, I think that wording is on the mark scheme is a bit off. I don't really agree with that. I would say you're better off describing that SOD's rate of conversion of superoxide into hydrogen peroxide is extremely high, and that the enzyme now becomes a limiting factor, which on the mark scheme, if you said SOD activity is high, that was actually credited. I think that would be a better phrase instead of catalase activity low. Is SOD activity may be too high. Question part 2 is scientists investigate the effect of hydrogen peroxide on sperm cells. This is a method they use, so incubate sperm cells with different concentrations of hydrogen peroxide for 2 hours, measure the motility of cells compared with normal sperm cells, measure the percentage of cells that have changes in composition of phospholipids in the plasma membrane. They used sperm samples from 10 different men attending a fertility clinic and calculated mean values. The results are shown in figure 21.2. Okay, so we've got motility compared to normal, with changed plasma membrane phospholipid composition as a percentage, and then concentration of peroxide on the x-axis. So here we can see that x denotes motility, whereas the dots denote cells with changed plasma membranes. Okay, and the question is that scientists concluded that hydrogen peroxide caused changes in the plasma membranes of sperm cells that reduces their motility. Evaluate this conclusion. So what we're looking for over here is higher peroxide, higher hydrogen peroxide results in changes to plasma membrane, and reduction in motility. Okay, so let's see if that phrase is translated up here. So higher hydrogen peroxide concentration. Yep, we could see that this is actually reduced because motility is the x, right? We could see that, so just comparing, so here's initial motility, decreases as the concentration increases, decreases slightly more, and then decreases more. So yes, there does seem to be this downward decrease, and that in relation to the plasma membrane, we see that as concentration of the peroxide increases, the cell membrane gets more or the plasma membrane gets more damaged. So yeah, we can actually see that. So we could put those statements down in support. However, there's no stats test. Also, sample's too small, 10 different men. So that would be evidence against the statements that the scientists made. Another conclusion that you could draw is the fact that over here, so for example, is some of the, actually not the bottom one, just the top one. So just this one, we could see the not standard deviation, the error bars, they overlap, which means that there's no significant differences between the means. So that could be something else. This does not really look to be an overlap. They're just about the same level. Actually, that one's slightly lower. Right, so that would be evidence for and evidence against. Part 3 is to suggest how hydrogen peroxide could affect the plasma membrane. So I believe it was mentioned earlier in the question that peroxide is extremely reactive somewhere. Yes, over here. So peroxide is an extremely reactive chemical. So what that would mean is that it has the potential to react with components in the cell membrane, namely proteins. It could damage or denature the proteins, disrupt the phospholipid bilayer. All of these things would make the membrane more permeable. Question 22, the last question on this paper, is a student investigates the effect of ethanol on membranes of beetroot cells at 25 degrees Celsius. They place equal masses of beetroot in tubes containing different concentrations of ethanol. They then use a colorimeter to measure the concentration of purple pigment that leaked out of the beetroot cells. The student replicates the experiment 5 times at each ethanol concentration and plots a graph of absorption in arbitrary units against concentration of ethanol. The student controls the mass of beetroot in this investigation. Identify 2 other variables that the student should control in this investigation. The other variables could be species of beetroot, for one, as well as the perhaps dimensions of the beetroot. So we're only investigating the effect of ethanol over here, and they've told us that temperature is constant, which is why temperature is not an answer. We know that the mass of beetroot is also constant, but what about the dimensions? If one beetroot piece has a larger surface area than the other, well, the one with the larger surface area would be more affected. So that could be one. Also, yeah, we're changing concentrations, but we could keep the volumes, so volume of ethanol, the same. So volume of ethanol, and other answers in the mark scheme included the same part of the beetroot, no skin on the beetroot. We said the species of the variety, age of the beetroot could be another one, time that the beetroot piece is spent in ethanol, as well as the volume of ethanol. Question B is the student's graph is shown below. Explain the shape of the graph. So on this graph, we can see that as ethanol concentration increases, absorption increases, but that's a description. So to explain it instead, you would say increase in ethanol concentration increases absorption because more ethanol would increase the membrane permeability. So that means more of the pigment can leak out. And there comes a certain point between, I would say 80 to 100, where there's no more pigment leaking out. So it would level off, the curve levels off as no more pigment is released. The very last question is a student repeats the experiment at a temperature of 30 degrees Celsius. Sketch the graph that you would expect the student to obtain. Answer on the graph itself. A higher temperature will make the phospholipid bilayer more fluid, which means more of the pigment can leak out, therefore absorption increases. So it would be like slightly higher, so towards the left of this, but the max absorption would be the same. So it's higher initially, so you need a lower concentration of ethanol to get a higher absorption, but both level off the same amount. Marking-wise, it is 1 mark for the curve being towards the left of the one that's already drawn. Second mark is that both of them level off the same place. And this brings us to the end of the question paper. I hope you found this useful. If you did, please give it a like and subscribe. Thank you for watching.